My name is Paul Earnshaw. I'm the Director of Vetter's Global Clinical Business Development Team, and I'd like to welcome you to our webinar, sorry. Over the next half hour, we'll introduce you to Vetter's dedicated clinical manufacturing sites in Chicago, Illinois, and Rankweil, Austria, and with the goal of helping you determine whether Vetter's capabilities are a good fit for your drug product requirements. I'm going to start with just a brief overview of Vetter. So Vetter is a private family-owned business with no products of our own, which means 100% of our efforts go towards supporting our customers. I think this differentiates us quite a bit from other CDMOs and allows us to operate independent of shareholders. I think additionally, our customers appreciate that we're not at risk of being acquired. That's something that has become pretty common in the industry. So Vetter currently manufactures over 115 commercial products at our 3 manufacturing facilities in Ravensburg, Germany. Across our organization, our 5 dedicated manufacturing facilities, we have 21 filling lines for vials, syringes, and cartridges. We have an outstanding international regulatory track record, and on average, a Vetter facility is inspected every other day, whether it's by a regulatory agency, a customer, or one of our internal QPs. So our organization is set up to support customers from preclinical through commercial, and that includes assembly and secondary packaging. Vetter is a very healthy organization with revenue in 2024 exceeding 1.1 billion euros. Being a private organization, it's allowed us to continually invest heavily in both existing and new filling lines. And this year, really excited to share that we exceeded 7,000 employees worldwide. We manufacture a diverse array of compounds covering numerous indications. This includes biologics, oligonucleotides, and peptides. Just to clarify, what we don't manufacture are high-potent compounds such as ADCs. We also don't manufacture live virus. So focusing a little more on our 2 dedicated clinical sites in Chicago, Illinois, and Rankweil, Austria, we have a total of 3 separate filling lines. In Chicago, we manufacture both liquid and lyo vials on one of our lines, and we have a separate line for syringes where we manufacture 1 ml long and 2.25 prefilled syringes. And in Rankweil, Austria, we manufacture both liquid and lyo vials. And we'll get into quite a bit more about this capabilities at the 2 facilities. Our early stage sites have supported over 140 different customers, headquartered in 21 different countries, and that includes 16 different states in the US. And this is my final slide before we dive into the sites specifically. We do support programs at our clinical sites from some of our larger pharma customers, but we're very proud of the fact that over 70% of our customers in Chicago and Rankweil have less than 200 employees. And I'm very excited to share that at our 2 dedicated clinical sites, we've supported over 280 customer audits. We manufactured over 380 unique compounds, and we currently have over 100 ongoing projects. So those were my final slides, just introducing Vetter. I'm going to go ahead and hand it over to my colleague, Mark Rockhurst.
Thank you, Paul. Great introduction to Vetter in general, and thanks for the high-level overview of our clinical sites. So what I'd like to do now is jump in and dive a little deeper into our clinical manufacturing site in Chicago. So diving a little deeper into Chicago specifically, our site was really created from the beginning and designed to deliver on customers' key drivers in early phase clinical manufacturing. So these are really things like speed. So drug product manufacturing is typically the last real step in the whole long process of manufacturing before going into key clinical trials for customers. So everything has been designed in our process, really from kickoff of a project to manufacturing to release to shipment, to reduce these timelines. Drug substance is also usually highly valuable and limited in quantity. So preservation of drug substance for manufacturers is another driver in all of our process design. And then ultimately, we're looking beyond the clinical manufacturing and really as true partners, looking at scalability and the eventual transfer to late phase commercial manufacturing and our partner lines in Germany. So if we look a little deeper into the flexibility, so our systems are built in a modular way, so we can compound anything from sub-liter up to 200 liters of drug substance here in Chicago. So smaller scale, we're compounding in bottles with stir bars. As we get larger, we're moving into mixing containers with impellers embedded in them. And then container types, anywhere from disposable bags, Pyrex bottles, a variety of aseptic connectors. So since we're dealing with so many different drug substance manufacturers, we're ready to work with different bags, bottles, connectors, really whatever drug substance manufacturers are providing. And then also a variety of different filters available to us. So here, looking at beyond the flexibility with the conservation of material, 100% of our product contact materials are single use. So there's a couple ways here where we're reducing cost, time, as well as preserving that drug substance. So we don't need to do product-specific cleaning validations. We don't have to order product-specific vessels, everything single use, single contact. So this is speeding things up, reducing material that's needed to do cleaning validations and reducing the timelines to get product-specific vessels. Again, really looking at the product conservation. The main things that tend to lead up to loss of material are things like the number of filters, the size of filters, and any in-process controls that are destructive. So here, in our processes, we really have a variety of different filters available to us. So we're very careful with filter sizing and selection to minimize that loss in the housings. Our machine is set up with a dose in and 100% inline weighing. So what that means is basically all the vials coming off of our vial line, we know are hitting the target volumes and weights. We don't have to do any in-process destructive IPC testing to make sure that our machine is drifting off of our targeted weights. And then again, minimizing the lengths and diameters of all of our tubing. On the right, you can see kind of a simple schematic of what our process looks like. So here on the left part in that box is what represents that final pooling container. So leading up to this, we've diluted, pooled, or compounded a powder API through a bioburden reduction filtration into this final compounding vessel in grade C under laminar airflow. From here, we're pumping with peristaltic pumps into our clean room through grade B into grade A environment, through 2 inline sterile filters into a break tank, which resides outside of the RABs. From here, we're pumping into the RABs, again, via peristaltic pumps into our 2-headed filling machine. Typical line losses we're seeing, again, really a lot of this depends on the filter choice, but anywhere from 50 to 150 milliliters of line loss during manufacturing. So we do have 2 clean rooms, 2 filling machines in Chicago. The first is an automated vial line. So this is using all ready-to-use pre-sterilized vials. We have liquid and lyo capabilities in Chicago. So the qualified formats, we're filling 2R, 6R, 10R, 20R, and 25R vials. The fill ranges, we filled anywhere between 0.3 up to 28.9 mLs for customers. This filling line runs maximum about 1,800 units per hour, which then dictates our fill time for about 15,000 piece max batch sizes. Lyo is dictated by the size of the lyo chamber. So here you can see the different breakdowns of max batch sizes for the different vial sizes. Okay, and then our other line is a Groninger syringe line. So here we're using pre-sterilized single chamber syringes. We're qualified and fill 1 ml and 2.25 ml syringes. These range anywhere from 0.1 up to 2.25 mLs in this line. Again, is a nest and tub system, so we do run a little quicker. So we're running 3,600 units per hour, which increases our batch sizes for the syringes. So for 1 ml longs, 25,000 units, and the 2.25, 20,000 units. We are able to do vacuum as well as mechanical stoppering. With the vacuum stoppering, it does reduce those line speeds a little bit, so the max batch sizes drop down a couple thousand pieces each. And then real quickly, just to talk about some of our utilities. So, Vetter really has taken everything in-house here, control of our own utilities. So we do have reverse osmosis water in place. We generate all of our own Wi-Fi, pure steam, as well as the Wi-Fi. We have two Wi-Fi loops, a cold loop and a hot loop. Again, 85 degrees and 25 degrees. Pressurized air as well as nitrogen battery on site. And I will also hit on everything, all critical components, systems, and equipment are on battery backup power, which then is also connected with a generator. So backup interruptible power for all critical components and instruments. And then finally, warehousing. So we do have a tremendous amount of warehousing for all of our materials and drug product, drug substance. So we have GMP storage, everything controlled by our SAP system. And again, room temperature, cold storage, as well as -20 and -80 capability for drug substance and drug product. I think from here, I will pass this off to Gareth and let him introduce you a little more about our site in Austria.
Thanks, Mark. Thanks for giving this an introduction to Chicago. And really, after Chicago put Vetter on the map as a manufacturer for early phase clinical material, it made our job in Austria very easy. When Vetter had the chance to come in 2021 and establish a site in Europe, all we had to do was copy the work and the process landscape that they put in place in Chicago and evolved over the last 10 years of success in manufacturing, and apply that to what we found in our building in Austria, in Rankweil. So I'll go through some of the details here. A lot of it is repeated because we like to have the same offering on both sides, but for the sake of completeness, we'll talk through each of them and then go into some of the unique things that we can both do. Okay. Our filling line is also from Groninger. It's a two-headed filler that utilizes also only single-use materials. Slight difference to Chicago, we process bulk vials here, so we go through a washing and depyrogenization process before going into the RAB system that we have. But very similar, all of our processes are set up to really optimize yield as much as possible. At this stage of development, typically drug substance is not available in sufficient amounts, so we need to really optimize things as much as possible. So all of our equipment has been selected to really get everything out of a fill as we can. So everything 100% single use, as Mark mentioned, to save time and expense on cleaning validation activities. And everything else is really identical to Chicago. We always have one eye also on that potential for success. Whenever your product can hit phase 3, we want to put processes in place that are easily transferable to that commercial scale later on. From a scale size, we're in exactly the same region as Chicago, so we can process from sub-liter volumes right the way up to a stop at 200 liters, which is where our carrier systems end. A range of different systems available through glassware, bag systems with those impellers, as Mark mentioned, if necessary, right the way from 10 up to 200 liters, covering our way across that. And we know things are very diverse. We get a lot of different diverse systems coming in, so we want to be able to handle all of them. So a number of different connection types are available. I think we're able to handle pretty much everything coming through, just as Chicago also. The slight differences in the pictures here. Our process steps, as Mark mentioned previously, one of the biggest killers for volume during processing can be that filter size. If you choose the wrong filter, you've got a massive holdup volume that can really kill a couple of 100 mils. So we are very careful in selection of that with you. A lot of different membrane types available, and we make sure to select the right housing to suit your product. Our process steps in Rankweil slightly different. Our break tank is located in grade A. So after our material is filtered and stored in a holding tank in grade C under laminar flow, tubing is passed through grade B into grade A, where the sterilizing filters are held, one or two, depending on your process needs. Goes through a break tank before going through those filling needles, and then going into the vials. In Rankweil, we also have that 100% inline weighing system, where vials are weighed, then filled, weighed again, to generate that fill weight data. The line recalibrates itself constantly, so we're always try to be as close to the target fill volume line as possible. Just due to the size of our line, our maximum line losses are slightly higher than Chicago, but we really do try to optimize those. Between 50 and 300 mils is where we're typically seeing our process line losses be, which I think is still a very acceptable level. On the next slide here, we have just an overview of our vial line and some of the numbers coming out of it. We do process liquid and lyophilized vials. The currently qualified vial formats are 2, 6, 10, and 20R. We are adding in 25R, and through the end of Q1 of 2026, that will be available as well. So we'd have all of those bases covered with different suppliers of glassware available on site. The line can fill very large batches, as you can see here. The maximum filling speed in that 2R vial format would be around 6,000 units per hour. We're typically running at about 20% of that for most of our customers, but we do have the option to go higher if larger batches are needed. The maximum batch size, as we said, dictated by the size of the lyophilizer. It is a commercial scale lyophilizer, so we can fill up to 60,000 2R vials within that. It's around 14.4 square meters. 10 shelves, so quite a lot of capacity there. Typically, we're filling individual shelves for the clinical need of our customers between 5 and 8,000 is our typical batch size there. In terms of utilities on site, we are a standalone facility also, so we generate all of our own water, for example, with a purified water system. With a very large tank that gives us enough capacity to meet our needs. And from there, we can go into clean steam or water for injection, again, for washing or sterilization of all of the parts that we need. Pressurized air with compressors on site to meet our capacity. Nitrogen, we have a battery system with sufficient backup to make sure we never run out. And we have a new generator on site as of this year that gives us an uninterruptible power supply with a generator not just for the critical systems, but it supplies the entire site with enough power within milliseconds if needed. In terms of storage and logistics, same thing as Chicago with the same storage conditions. We have room temperature storage typically for all of the components and starting materials that we have. Cold storage, usually for finished goods, prior to shipment and samples, and then frozen storage for -20, where we're seeing a lot of oligos being stored, and -80 primarily for biologic storage. And as in Chicago, also all of our material movements are covered by SAP, and material status is readily available and visible within that SAP system. So those are some of the site specifics. I'm now looking at some things you'll see that are common among both sites, which are key to our process. The first up, very, very important for our timelines, are the drug substance requirements that we have. So for incoming goods testing of drug substance, we do not require identity testing if some prerequisites can be met. Namely, if we have tamper-evident sealing on the shipment, if there's unique identifiers on those, so we can clearly say that this material was not tampered with during transport. And also if the COA contains information around identity, bioburden, and endotoxin, we can forgo that identity testing requirement. And other than that, we don't really have any hard needs for our incoming goods inspection. All of them are really guided to maintain our clean room security and clean room safety, being a multi-product site. So besides identity, bioburden, and endotoxin, we just really need information around viral safety steps or mycoplasma absence steps that have been taken with your drug substance to make sure we're not contaminating our site or our facility. We can have a number of different container types. I think we've seen more or less everything in terms of different container types coming through, and the storage conditions that we have are tailored really to meet your needs for your material. Feder does have a wide range of suppliers and excipients available, but if you have specialty materials of a certain quality, a certain grade from a certain supplier that you need for your formulation, we have the option that customers can provide that material to us. There is a bit of a longer time required for that as ID testing needs to be performed, but it's certainly an open topic that we can easily cover within the project work. Looking now at the back end of our process steps, in terms of testing, both sites are set up to perform a wide range of different testing for your product. Simple compendial testing, but also product-specific options are available either on-site in Chicago or Rankweil, but also centrally in our Ravensburg headquarters, where we have a wide range of different QC methods available, and we're happy to talk about transfer of those methods to us. Stability storage is also something we can support with using our facilities in Ravensburg that have access to all of the qualified ICH storage conditions necessary, and also testing can be performed there, and they're very experienced in that regard. Microbiome-wise, obviously we perform sterility testing, endotoxin and bioburden testing there for most batches. We can support with a number of different options there. And all of our environmental monitoring is also performed in-house. We have quite a large library of strain identification exercises performed, so we're able to identify everything there. So how do we actually operate and process? The customer-facing process is pretty much the same, independent of site. Our first, well, the key portion of our project work is really generating a process development specification, our leading document, our product Bible, if you like. And to get to that document, we'll start a process with really understanding your product. So we'll assemble a core team on site with subject matter experts from a number of different departments, really start asking you a lot of questions about your product, about experience with that product and so on, really identify where that fits into our process landscape, and then put together a development plan. If that requires small scale process development work to characterize processes more closely before going into manufacturing, that is something we can support with our development lab in Ravensburg. Or we put risk assessments in place to make the justified and informed choice about our process steps that we then capture in a process development specification that gets jointly approved between both teams and covers things like the process steps, sampling, and all of those specifications around hold times, for example, that you might have. Our project teams look very similar for all of our projects. We have a dedicated, very experienced project manager. Mark and I are in the happy situation to have a great team of project managers under us, and they'll put together the team with folks from quality assurance, quality control, process development, which we call technology and process transfer, but also supply chain to support with import, export, and sample shipment pieces. And around that, within Feder's network, we have a wide range of different functions available, if necessary, that we can pull in as needed to give support for specific issues we may be having, it's a great position to be in. That's kind of the end of our brief overview of our early phase clinical manufacturing capabilities. I'll hand over to Paul to give us a short wrap up. Thanks, everyone.